1. DNA replication occurs in the 5 to 3 direction. Which of the following best describes this process? Leading strand synthesis occurs in the 3 to 5 direction, while the lagging strand is synthesized in the 5 to 3 direction. B. Leading strand synthesis occurs in the 5 to 3 direction, while the lagging strand is synthesized in the 3 to 5 direction. C. Both the leading and lagging strands are synthesized continuously in the 5 to 3 direction. D. DNA replication occurs in the 5 to 3 direction in prokaryotes, but in the 3 to 5 direction in eukaryotes. E. Both the leading and lagging strands are synthesized continuously in the 3 to 5 direction. Answer: B. Leading strand synthesis occurs in the 5 to 3 direction, while the lagging strand is synthesized in the 3 to 5 direction. Explanation: DNA replication occurs in the 5 to 3 direction for both strands, but the leading strand is synthesized continuously in this direction, while the lagging strand is synthesized discontinuously in the 3 to 5 direction in Okazaki fragments. 2. Which of the following enzymes is responsible for unwinding the DNA double helix during DNA replication? A. DNA polymerase, B. Helicase, ligase, D. Topoisomerase, E. Primase. B. Helicase. Explanation: Helicase is the enzyme responsible for unwinding the DNA double helix at the replication fork, creating 2 single-stranded templates for DNA synthesis. The other enzymes listed play different roles in replication, such as polymerizing DNA or managing DNA tension. 3. In which of the following genetic conditions is there a deficiency of the enzyme helicase leading to problems with DNA replication? A. Bloom syndrome, B. Xeroderma pigmentosum, C. Fragile X syndrome, D. Cystic fibrosis, E. Huntington disease. Bloom syndrome. Explanation: Bloom syndrome is caused by mutations in the BLM gene leading to a deficiency in helicase function. This results in genomic instability and defective DNA replication. Xeroderma pigmentosum involves defective DNA repair mechanisms, while the other conditions do not involve helicase deficiencies. Nata 4. Which of the following best describes the origin of DNA replication in eukaryotic cells? A. A single consensus sequence, typically AT-rich, is the origin of replication. B. Replication occurs at multiple origins of replication across the genome. C. Only one origin of replication is required for the entire genome. D. The origin of replication is located in the Tata box, a region found in promoters. E. Eukaryotic replication starts at the origin of replication, which is always located at the 3' end of the DNA sequence. Answer: Replication occurs at multiple origins of replication across the genome. Explanation: In eukaryotes, replication occurs at multiple origins of replication, each with AT-rich sequences. This contrasts with prokaryotes, where a single origin of replication is typically sufficient for the entire genome. 5. Which of the following best characterizes the DNA replication process in prokaryotes compared to eukaryotes? A. Prokaryotic replication is less complex and involves fewer enzymes than eukaryotic replication. B. Prokaryotic replication occurs in the 3 to 5 direction, while eukaryotic replication occurs in the 5 to 3 direction. C. Prokaryotes use multiple origins of replication, whereas eukaryotes have a single origin. D. DNA replication is discontinuous in prokaryotes and continuous in eukaryotes. E. Prokaryotes do not require helicase for DNA unwinding. A. Prokaryotic replication is less complex and involves fewer enzymes than eukaryotic replication. Explanation: Prokaryotic DNA replication is simpler than eukaryotic replication, with only one origin of replication and fewer enzymes. Eukaryotic replication is more complex, involving multiple origins and more regulatory steps. 6. Which of the following regions in DNA is most likely to be involved in the origin of replication? A. GC-rich sequences, B. Tata box regions, C. Poly-A tail regions, D. Exonic regions, E. Intronic regions. Answer: Tata box regions. Explanation: The Tata box is an AT-rich sequence found in both promoters and origins of replication. It plays a key role in initiating DNA replication and transcription. GC-rich sequences are more often involved in stable DNA binding, while the poly-A tail and exons introns are related to RNA processing rather than replication. These questions cover